Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is linkage and crossing over. This video is from CBSE Class 12th, Chapter 5th, Part 3rd of the Genetic series. Before this, we discussed about the chromosomal theory of linkage. So here, the first topic, that is linkage. So what is linkage? Linkage is the close association of genes or other DNA sequences on the same chromosome. The two closer genes are to each other on the chromosome, the greater probability that they will be inherited together. So in case of linkage, a recombination frequency of 50% means that genes are unlinked. There are two ways in which genes may be unlinked. They may be on the separate chromosome or they may be far apart on the same chromosome. So here in this picture you can see linked genes which are closely associated, and in the second and third chromosome which shows unlinked genes where even genes are present on different chromosomes or on the same chromosome genes are far apart from each other. The genes located in the same chromosome are called linked genes, and those present in different chromosomes are termed unlinked genes. So in this picture, both chromosomes shows the unlinked genes because the genes are present on the different chromosomes. And here in second picture, it shows gene second, gene third and gene fourth, a set of genes at different loci on the same genes that except for crossing over tend to be inherited together. The term linkage was coined by T.H. Morgan. He carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. Genetic linkage analysis is a powerful tool to detect the chromosomal location of disease genes. It is based on the observation that genes that reside physically close on a chromosome remain linked during meiosis. So, the principle of linkage explains why in the fruit fly the red eyes and grey body occur in the same individual and why it go together in the gamete and in the next generation. Now, the demonstration of linkage, and here the first one is unlinked genes. The genes located in different chromosomes assort independently, give a dihybrid ratio of 9:3:3:1 and show a test cross ratio of 1:1:1:1. So here you can see there are two parents, capital A, capital A, capital B, capital B, and small a, small a, small b, small b, and both are homozygous. Then at the time of gamete formation, independent assortment occur, and after this in F1 progeny, heterozygous condition develop, that is capital A, small a, capital B, small b. After the test cross, here the heterozygous dominant, that is capital A, small a, capital B, small b, cross with the homozygous recessive, and after that in the next, that is test cross progeny, there are four types of individuals in equal proportions formed. So here you can see the ratio of 1:1:1:1 and it shows the result of the independent assortment of unlinked genes during gamete formation. Now, the linked genes. The genes located in the same chromosome usually do not assort independently, but remain together in the gametes and the offsprings, give a dihybrid ratio of 3:1 and shows a test cross ratio of 1:1. So in this picture, here the two non-crossover gametes, that is parental combination, and two crossover gametes, that is recombinant gametes, it shows 2:2 ratio, means 1:1 ratio, and so the linked genes inheritance. Now, here, the progeny of above dihybrid test cross comprises only two types of individual, that is produced in equal proportion. So, both are parental forms and no recombinants have been formed here. So, because of this, the linked genes come together and stay together in the next generation. And this is the behavior of linked genes in their assortment in gametogenesis. Here you can see that is unlinked genes are that type of genes which are inherited independently, as they are either located far apart on the same chromosome or on different chromosomes altogether. It means that genes will follow the general rules of Mendelian genetics. So here you can see the normal dihybrid ratio, that is 9 is to 3 is to 3 is to 1, and test cross ratio, that is 1 is to 1 is to 1 is to 1. While in case of linked genes, that is, you can see here, that is monohybrid pattern, the linked genes that are linked likely to be inherited due to their loci being close in proximity. And this basically means that if two genes are located close together on a chromosome, then they are likely to be paired down to the offspring together. And this is easy to understand if you think about the physical location of the genes on a chromosome of two genes are close together on one leg, and that the chromosomes, they are likely to be pulled to the same side during meiosis. Moreover, if crossing over does occur, then genes that are close together are likely to be crossed to another chromosome together. So this is the case of linked and unlinked genes. Now, the chromosomal theory of linkage, in which the principle of linkage was first discovered by Betson and Punnett in 1906 in sweet pea. It was put forward as a regular concept by Morgan in 1910 from his work on fruit fly, that is Drosophila melanogaster. Morgan and Castell formulated the chromosome theory of linkage with the following postulates, in which the first one is genes lie in a linear order in the chromosomes. The genes which show linkage, that is stay together in the hereditary transmission, are located in the same chromosome. Genes tend to stay in the parental combination except for occasional crossing over. The distance between the linked genes in the chromosome determines the strength of linkage. The closely located genes show stronger linkage than the distant genes because the latter are more likely to undergo crossing over than the former. After this, kinds of linkage. So there are two kinds of linkage, in which the first one is complete linkage. The genes closely located in the chromosome show complete linkage, as they have no chance of separating by crossing over and are always transmitted together to the same gamete and the same offspring. Thus, the parental combination of traits is inherited as such by the young one in case of complete linkage. Here, the parental combination of traits is inherited as such by the young one in case of complete linkage. Now, here you can see a dihybrid test cross showing complete linkage. And here, a double dominant female Drosophila homozygous for red eyes and normal wing, that is a wild type, is crossed with a double recessive male Drosophila homozygous for purple eyes and vestigial wings, which you can see in the picture. The F1 flies are all heterozygous, red eyes and normal wings. F1 hybrid female flies is then test crossed with a double recessive male having purple eyes and vestigial wings. So in F2 comprises only two types of flies, that is red eyes normal wings and purple eyes vestigial wings in the ratio of 1:1. So no recombinant types are formed because linkage is complete and no crossing over occur. Now here you can see incomplete linkage. So the genes distantly located in the chromosome show incomplete linkage because they have a chance of separation by crossing over and of going into different gametes and offsprings. So in this picture you can see incomplete linkage in maize seed, that means Gia maize. A case of incomplete linkage here, and here the between the alleles for color and shape of seed has been observed by Hutchinson. When a maize plant with seeds having 9 colored and full endosperm, that is capital C capital S and capital C, capital S, which you can see in the picture, that is colorful seed, crossed with another plant having recessive alleles for colorless shrunken seeds, that is small s, small s, small s, small s. So the F1 heterozygous are found with the phenotype of colorful and genotype of capital C, capital S, small s. So when F1 hybrid test cross with double recessive parent, that is small c, small s, small c, small s, four classes of descendants are obtained instead of two. So here the test cross results are clearly showing that the parental combination of alleles are those expected from complete linkage and appear in 96% cases, and the other two are the new combinations and appear in 4% cases. So then in 4% cases crossing over has occurred between the linked genes, and because of this it shows the incomplete linkage. Now here you can see that is a Betson and Punnett experiment showing incomplete linkage and crossing over. Here an early case of incomplete linkage which allows the separation of linked genes was noted by Betson and Punnett in sweet pea, that is Lathyrus odoratus, in 1906. They crossed a double dominant sweet pea plant homozygous for blue flowers and long pollen grains with a double recessive plant homozygous for red flower and round pollen grains. So the F1 plants were heterozygous for blue flower and long pollen grains. So because of this, the F1 dihybrid plant was test crossed with a double recessive parent, and the progeny of this cross, instead of the normal dihybrid test cross ratio of 1:1:1:1:1, showed an unexpected phenotypic ratio of 7:1:1:7. So, in a dihybrid test cross involving independent assortment of two genes and the frequency of four phenotypes in 25% each. So, the total frequency of the parental forms 50% is equal to the total frequency of recombinant, that is also 50%. Now, what is the difference between the crossing over and linkage? So, here you can see in case of crossing over, it leads to separation of linked genes, while in case of linkage, it keeps the genes together. In crossing over, it involves exchange of segments between non-sister chromatids of the homologous chromosomes, while in linkage, it involves individual chromosomes. After this, in case of crossing over, the frequency of crossing over can never exceed 50%. While in case of linkage, the number of linkage groups can never be more than haploid chromosome number. In crossing over, it increases variability by forming new gene combinations, while the linkage, it reduces variability. Now, the factors affecting linkage. In this, the first one is, which you can see here, that is the age. So, with advancing age, the chances of crossing over lessen and increasing the strength of linkage. While in case of temperature, rise in temperature increases the chances of chiasma formation and decreasing the strength of linkage. In case of X-rays, exposure to X-rays reduces the strength of linkage. So here one thing you have to know, that what is the linkage group. So all the genes in a particular chromosome form a linkage group, since the genes in a particular chromosome have their alleles in the corresponding homologous chromosome. A pair of homologous chromosomes are said to form a single linkage group. Like as an example, you can see here, that is Drosophila melanogaster has 4 linkage groups, 3 large and 1 small. That corresponds to the 4 pairs of chromosome, 3 long, 1 short it possesses. While in case of man has 23 linkage groups and 23 pairs of chromosomes. among plant, like in case of the garden pea, has 7 linkage groups and 7 pair of chromosomes, and maize has 10 linkage groups and 10 pair of chromosomes. So these are the linkage groups. Now the significance of linkage. So it greatly reduces the chances of the formation of new combinations of genes in the gametes. It helps in maintaining the valuable traits of a newly developed varieties. Linkage disallows the plants and animals breeders to combine all the desirable traits in a single variety. The fact that there are as many linkage groups as the chromosome pairs in an important evidence for the location of genes in the chromosomes. After this, next is sex linkage. So here you can see the genes along with the sex controlling genes from generation to generation and are therefore called sex linked genes. The characters controlled by the sex linked genes are termed sex linked characters. Inheritance of sex linked genes or traits are known as sex linked inheritance. The location of sex linked genes in the chromosomes is called sex linkage. If sex-linked genes are located on the X chromosome are called X linkage, and if the sex-linked genes are located on the Y chromosome are called Y linkage. After this, now the inheritance of sex linkage. So the sex linkage was first discovered by Morgan in 1910 in the fruit fly Drosophila melanogaster. Here, XX and XY sex chromosomes in the female and males respectively present. Its eye color is sex-linked. Allele of the eye color gene is located on the X chromosome and there is no corresponding allele in the Y chromosome. The male expresses a sex-linked recessive trait even if has a single gene for it, whereas the female expresses such a trait only if it has 2 genes for it. The normal eye color is red and it is dominant over the mutant white eye color. Now, the following crosses illustrate the inheritance of X-linked eye color in Drosophila. So now here, the inheritance of X-linked eye colors in case of Drosophila, and here the first one, that is the red-eyed female and the white-eyed male. So here you can see that is if a homozygous red-eyed female fly is mated with a hemizygous white-eyed male fly, all the F1 flies irrespective of their sexes are red-eyed. So when the red-eyed male and female flies of F1 are intercrossed, the F2 flies are in the ratio of 2 red-eyed female to 1 red-eyed male to 1 white-eyed male. So here, thus the red-eyed and white-eyed flies are in the ratio of 3:1 in F2 generation. And if here you can see that is XR represents a gene for red eye and X small r represent the white eye color. So this shows the inheritance of X-linked eye color in case of Drosophila. So these all the classes that are recessive X-linked that trait from the criss-cross inheritance, that is transmission from the father to the grandson through the daughters, and latter are called carriers because they have a trait but do not express it. So it shows the criss-cross inheritance. Now here the second, that is white-eyed female and red-eyed male. So here you can see the cross, and here in this cross a homozygous white-eyed female fly is mated with a hemizygous red-eyed male fly. This cross is reciprocal of the previous cross and should yield the same result, but in its F1 progeny all the males are white-eyed and all the females are red-eyed. This result shows that eye color and sex are linked. When a red-eyed female fly and a white-eyed male fly of F1 are intercrossed, F2 progeny comprises approximately equal numbers of red-eyed females, white-eyed females, red-eyed males and white-eyed males. So, the above cross shows that the father transmits its dominant sex-linked trait, that is red eyes, to his grandson as well as granddaughter through daughters. So, it shows the pattern of inheritance of eye color trait that is exactly parallel the behavior of the X chromosomes carrying the alleles of the eye color genes, and this proves that the eye color genes lie in the X chromosome. So this is the inheritance of X-linked eye color in Drosophila, and it shows the ratio of 1:1:1:1. Now the characteristic of sex-linked inheritance. It is a criss-cross. Mothers passes the alleles of the traits to both daughters and sons. Only homozygous females express an X-linked recessive traits. Heterozygous females are carriers of X-linked recessive traits and themselves do not express the traits. Any male having an X-linked recessive allele expresses the traits as his Y chromosome and has no counteracting allele. The disorder causing X-linked recessive genes express more often in the males than in the females. Females homozygous for an X-linked recessive trait transfer the trait to all the sons, and most of the sex-linked traits are recessive. So in main, 50 sex-limited, that means linked traits discovered, and these includes hemophilia, that red-green color blindness, etc. So what is the importance of the sex-linked inheritance? The knowledge of criss-cross inheritance can help us to predict the transmission of sex-linked disorder to the future offspring. And it also proves that genes lie in the chromosomes. Now, the sex-limited characters. So, certain genes produce characters in one sex only, but not in both, though they are present in both the sexes. Such characters are called sex-linked characters. Like example, bright plumage in some male birds and the milk secretions in case of female mammals. Second is sex-influenced traits. The autosomal traits in which the dominant expression depends on the sex chromosomes of the individuals are called sex-influenced traits. And here the example, that is the abnormality called hair lip, in more common and more severe in males than in females. So the pattern baldness in men is also a sex-influenced trait, and in this the hair loss typically occurs on the top and front of the head. In case of female, the pattern baldness is thinning occurs on the top and crown of the head. This thinning in women often starts as a widening of the center hair part that leaves the front hairline unaffected. So this is the sex-limited characters and sex-influenced traits. Now, crossing over. In 1909, the Belgian cytologist Janssen observed that chiasmata formation during the prophase of meiosis first. In 1910, Morgan found that chiasmata formation leads to crossing over of alleles by breakage and reunion of the homologous chromosome. So, the crossing over is the mutual exchange of the corresponding segments of the adjacent parental and maternal chromatids of the synapsed homologous chromosomes in the packaging of meiosis first, producing new combination of genes. The chromatids resulting from the interchange of segments are known as the crossovers or recombinants. The chromatids that remain intact are called the non-crossover or parental chromatids. So this is the crossing over, and here that is process that shows the non-sister chromatids for chiasma and the crossover occur and form the new recombinants. So this shows the variation and the formations of new combinations just because of crossing over in the non-sister chromatids. Now, mechanism of crossing over. And here in this picture you can see the whole process, that is showing the crossover, and it produces 50% parental and 50% recombinant chromatids. So the process of crossing over occurs by a mechanism called breakage and reunion theory of Darlington in 1937, and it comprises 4 steps, in which the first one, that is known as synapsis. So, the homologous chromosomes come to lie close together in pairs in the zygote substage of the prophase of meiosis first. And here, their pairing is called synapsis. The paired homologous chromosomes are called bivalents, and their chromatids are not visible at this stage. Now, the second stage, that is known as tetrad formation. Here, the chromatids of each synapsed chromosome slightly separate and become visible in the Pachytene substage of the prophase of meiosis. A group of 4 homologous chromatids is called tetrad, and the crossing over occurs in the 4-stranded or tetrad stage. After this, now the crossover occurs. So, the crossing over also occur in the Pachytene substage, and the adjacent non-sister chromatids break and homologous sites mutually exchange small corresponding segments and rejoin. The process leads to physical exchange of segments and is so exact that neither chromatids gains or lose any genes. After this, the completion of crossing over, the synaptic forces end and the homologous chromosomes move apart. So, the site where the crossing over occurs are called gamete. Therefore, the above explained mode of crossing over is sometimes known as gamete-type hypothesis. So, this is the complete process, that means mechanism of crossing over. Now, here the crossing over leads to recombination of genes on the chromosomes, and the point of crossover if is visible for a cross-sebbed gamete. The exchange is usually reciprocal, and the exchanging segments of the 2 chromosomes are of similar size. So here the 2 homologous non-sister chromatids pair up with each other and exchange different segments of genetic material to form 2 recombinant chromosomes, sister chromatids. So this is the chromosomal crossing over in case of meiosis. Now the result of crossing over. So at first here in this pic, you can see the crossing over is the exchange of genes between 2 chromosomes resulting in non-identical chromatids that comprises the genetic material of gametes. And this process results in the millions of sperms or eggs that are produced by an organism, and each being different from one another. So the result of crossing over is each chromatid ends up in a separate gamete, 2 of which have the parental genotype and 2 the recombinant genotype. So this is the result of crossing over. Here you can see the linkage and crossing over are antagonistic to each other. The former keeps the genes of a chromosome together, the latter separates them. So if linkage and crossing over have equal chance of occurring, the crossing over will take place in 50% of cases and will result in the formation of 4 types of gametes with equal frequencies. That is, AB is 25%, A small b is 25%, small a capital B is 25%, and small a small b is also 25% from a heterozygous individual. And this frequency is same as for the 2 genes located on separate chromosomes. Thus, the genes may show independent assortment in spite of their location in the same chromosome. Hence, the independent assortment of genes can occur under 2 conditions. First one, when the genes lie in different chromosomes, and second, when the genes lie in the same chromosome but are far enough to get separated by the crossing over in all cases. So it shows without the crossing over, the four daughter cells below would have no genetic recombinations. Now, types of crossing over. So here you can see in the picture that according to the number of chiasmata of the homologous chromosome, crossing over is of following types, in which the first one, that is, in first case no crossover occur, that means there is no recombinations form. While in case of second, here that is the single crossover, here only one chiasma is formed between the non-sister chromatids. As a result, single crossing over occur and the two parental types and the two recombinant types form as a resultant. While in case of double crossover, the two chiasmata are formed and hence the two crossing over occur at 2 points in the same tetrad, and this type of crossing over is less frequent. After this, now here you can see there are two types of cross, that is 3-stranded and 4-stranded. Here in both cases these are known as the multiple crossing over. In this there are more chiasmata or forms, that means 3 or more than this, and here the corresponding to the number of chiasmata crossing over occur through this type does not occur frequently. So these all show the types of crossing over and its recombination frequency here. Now, what is the frequency of crossing over? So the frequency of crossing over is higher than the observed frequency of recombination of the traits in the offspring, because the occurrence of crossing over is indicated by the new combinations of traits in the offspring, and the two or more crossovers may take place simultaneously between the same chromatids of a tetrad. Now, the factors affecting crossing over. So here you can see that means loci on the chromosome, that is different places on a same chromosome, and it shows the different type of trait formation. So each genes are responsible for the formation of a new character, a new organ or new structure. Now the first factor that affect the crossing over is distance. So the frequency of crossing over of genes is directly proportional to the distance between them in the chromosome. So the more distant two genes are in a chromosome, the greater opportunity they have for crossing over. The closer two genes are in the chromosome, lesser chances they have for the crossing over. Second is age. Crossing over frequency generally decreases with advancing age. Next is X-rays. Exposure to X-ray increase the frequency of crossing over. Next is sex. Crossing over has not been reported in male fruit fly. Male mammals have less frequent crossing over. Next is temperature. Variation in temperature increases crossing over frequencies. Next, location. Location of genes near the ends and centromere reduces the frequency of crossing over. Next is chemical. Many chemicals found in the food are known to alter crossing over frequency in animals. And the last is interference. One crossover lessens the chances of another crossing over to occur near it. So these are the factors that affect the crossing over. Now, the significance of crossing over. It is an important factor in sexual reproduction. The variation introduced by crossing over forms raw material for evolution to occur, producing new species. So, the crossing over results in the numerous new combinations that can be acted upon by the natural selection. Crossing over has helped in establishing the concept of linear arrangement of genes. The frequency of crossing over helps in the mapping of the chromosomes, that is, determining the loci or location of the genes in the chromosomes. And the last, that is, it provides useful combinations of the traits to plants and animal breeders for producing new varieties. So because of this, selection of useful recombinants of traits by the geneticist has brought about green revolution in our country and is now heading for the white revolution. Now, the sex determination. So, sex chromosomes. What are the sex chromosomes? So, in case of diploid organisms with separate sexes, a specific pair of chromosomes determine the sex of the individual are called sex chromosomes or allosomes or heterosomes. All other chromosomes are autosomal chromosomes or autosomes. So, the two members of each pair of homologous autosomes are similar in size, but the sex chromosome are not similar in shape and size. So here you can see a sex chromosome in case of mammals, birds and Drosophila. Now here the dissimilar sex chromosomes, in which there are four conditions for the sex chromosome, and the first one is XXXY condition. So, in mammals, including man and most of the insects, including fruit fly, one sex chromosome is smaller than the other in the males. The larger one is known as the X chromosome and the smaller one is known as Y chromosome. In case of the female, these animals have a pair of X chromosome. Thus, the condition in male may be briefly expressed as XY, and then in case of the female XX. So, the similar and dissimilar sex chromosomes of the females and males are described as homomorphic and heteromorphic respectively. Now, second is XXXO type of sex chromosomes. In certain insects such as cockroach and some roundworms, the Y chromosome is missing so that the male has only one sex chromosome. And the condition in the male is XO. O means absence of one sex chromosome, and in the female it is XX. Third case, that is ZWZZ type. In male vertebrates and some insects, the female has heteromorphic sex chromosome and male has homomorphic sex chromosome. So these conditions are briefly indicated ZW, ZZ respectively, and these letters are used to avoid confusion with XX, XY condition. Last is ZO, ZZ type. In some butterflies and moth, the W chromosome is lacking, so that the condition in the female is Z0 or ZO, and that in the male ZZ condition. Now, after this, the mechanism of sex determination. So, the mechanism that fixes the sex of an individual as it begins life is called sex determination. Now here there are different type of mechanism, that is genetically controlled sex determination mechanism, metabolically controlled sex determination mechanism, hormonally controlled sex determination mechanism, and environmentally controlled sex determination mechanism, in which the first one here is, that is genetic or chromosomal sex determination. So here you can see that is sex determination chart, and every animal have different types of chromosomes, like here type, example, male, female, homogametic and heterogametic are given below, and you can find out that in which type who is homogametic and who is heterogametic. So here you can see there are two heterogametic males and the two heterogametic females. So in case of heterogametic males, there are two subtypes. First one is XXXO condition, where the sex chromosome in case of female is 2X chromosome and in case of male 1X chromosome. And one X chromosome or Y chromosome is lacking. And the examples are plants like Vallisneria, asparagus, bugs and grasshoppers. While in heterogametic male condition, second subtype is XXXY condition, where the females are 2X chromosomes and the males have X and Y chromosome. It occurs in man, other mammals, Drosophila, certain angiosperms, etc. In case of heterogametic female, There are two conditions also, ZO-ZZ type, where the females have one Z chromosomes and in males have two Z chromosomes. And the examples are moths and butterflies. While in second subtype, that is ZW-ZZ type, here the females have two ZW chromosomes and the males have two Z chromosomes. And the examples are gypsy moth, fishes, reptiles, birds, and mammals. So these are a complete description about the type, subtype, sex chromosome, and the case in which organism this type of sex determination present. Now, the first, that is XXXY type of sex determination. So in this case, you can see here, the sex is determined in mammals including human beings and certain insects such as fruit fly in the same manner. In both, that is human beings and fruit fly, the females have similar sex chromosomes, that is XX, and the males have dissimilar sex chromosome, that is X and Y. And here, the Y chromosome is shorter than the X chromosome in humans and is hooked in fruit fly. Though dissimilar, the X and Y chromosomes synapse in meiosis, segregate and pass into different gametes. Thus, in humans, the female is homozygous and male heterozygous for the sex chromosome. So in this picture, you can see the human beings have 46 chromosomes and these are distinguishable into one pair of sex chromosome and 22 pairs of homologous autosomes. During maturation, the sex chromosomes segregate freely like the autosomes. With the result, the ovum contains 22 autosomes and an X chromosome, whereas a sperm contains 22 autosomes and either an X or a Y chromosome. So, in this picture you can see the sex determination in Drosophila and the eggs are alike, each with X, and the sperms are of two types, that is X or Y. So female is said to be homogametic as it produces similar eggs and the male is said to be heterogametic as it produces two types of sperms. This condition is called male digamety. And the Y condition containing sperms and the X containing sperms are respectively called androsperms and gymnosperms. So the two are produced in equal proportions and the sex is determined all the time of fertilization by the kind of sperm that fuses with the ovum. So the egg with X chromosome fertilized by a sperm with X chromosome will become a zygote with XX chromosome, and if the zygote will and this condition zygote will develop into a female, and if the egg having X chromosome fertilized by a sperm having Y chromosome will become a zygote with XY sex chromosome and the zygote will develop into a male, and the Y chromosome is often called androsome and its condition in the zygote produces a male. So this is the XXXY type of sex determination. Now the second, that is XXXO type of sex determination. So in this condition, plants like Vallisneria and insects of order Hemiptera and Orthoptera, females have two X chromosomes and the male have only one X chromosome. So the male produces two types of sperms, half with X and half without X. So the presence of one unpaired X chromosome determines the masculine sex characters. So this is the condition of XO system of sex determination, which you can see here. That is, the female has two homomorphic sex chromosomes, that is XX, and is homogametic. It produces similar eggs and each with X chromosome. The males had one sex only and it is heterogametic. It produces two types of sperms. So the gymnosperms with X and androsperms without X chromosome. Fertilization of an egg by X bearing sperm yield female offspring and by no X sperms yields male offspring. Thus, the sex is determined at the time of fertilization as in XXXY type and the sex ratio is 1:1. So in this, Calvin Bridges demonstrated that in Drosophila, sex determining factor is a ratio of number of X chromosome to the set of autosomes. Therefore, Individuals having X by A ratio of 1 is to 1.0 are females and fertile, and examples are 2A plus XX or 3A plus XXX condition. If X by A ratio exceeds 1.0, metafemales are produced which are weak and infertile. X by A ratio of 0.5 is necessary for the male sex determination. And when X by A ratio decreases below 0.5, infertile metafemales are produced. Bridges also demonstrated that fruit flies having X by A ratio between 0.5 and 1.0 revealed a number of morphological and sexual abnormality and were referred to as intersexes. So these findings suggested that in case of Drosophila, male development factors are localized on autosomes. And here, this mode of sex determination is termed as genic balance theory of sex determination. Now, the next is ZW-ZZ type of sex determination. So, in this condition, this type of sex determination pattern is common in case of birds, butterflies and moth. It was discovered by J. Seller in 1914 in moth, and it is the reverse of XY-XX system. Here, the female is heterogametic, so ZW, but the male is homogametic, that is ZZ. Female produces two kinds of X, Z and W in equal proportion, while all the sperms are alike and each carrying a Z chromosomes. So here the fertilization of an egg with the Z chromosomes by a sperm with Z chromosomes gives a zygote with ZZ chromosome, and this zygote develops into a male. While the fertilization of an egg with YW chromosome by a sperm with Z chromosomes yields a zygote with ZW chromosome, and this zygote produces a female. The offspring have sex ratio of 1:1. So here you can see how the male and females are formed. In case of male, autosomes plus ZZ chromosomes, while in case of female, autosomes plus ZW chromosomes. Now the next is ZO-ZZ type of sex determination. And in this is found in certain moth and butterflies. Females possess single Z chromosomes but in its body, hence is called ZO, that is heterogametic, produces two kinds of X. And male possesses two Z chromosomes referred as ZZ and produces one type of sperm. So the sex of offsprings depends upon the kinds of egg. So here you can see the fertilization by a sperm with Z chromosome. The Z containing eggs give rise to a male offspring, that is ZZ, and the Z lacking egg produces a female offspring, that is ZO condition. Now the fifth is, that is haploid diploid type of sex determination, that is haplodiploidy. So Hymenopterous insects such as bees, wasps, sawflies and ants show a unique phenomena in which an unfertilized egg develops into a male and a fertilized egg develops into a female. So here you can see the female is diploid, that is 2N condition, and the male is haploid, that is N condition. So eggs are formed by meiosis and sperms by mitosis. Fertilization restores the diploid number of chromosomes in the zygote, which give rise to the female. If the egg is not fertilized, it will still develop but into a male. Thus, the sex is determined by the number of chromosomes, and it shows the sex determination in case of honeybee, that means haplodiploid type of sex determination. Now, what is the differences between the male heterogamety and female heterogamety? In case of male heterogamety, male produces two different types of sperms, while the female produces only one type of gametes. While in case of the female heterogamety, female produces two similar types of gametes, while the male produces only one type of gametes. In male heterogamety, XY or XO type or two types of heterogamety present, while in case of female heterogamety, ZW type or one type of heterogamety present. In male heterogamety, examples are like grasshopper, bugs, etc. While in case of female heterogamety, the examples are like birds, reptiles, fishes, etc. So, after this, the second mechanism of crossing over, that is the environmental sex determination. So in some animals there is environmental sex determination of sex, like in case of Bonellia, a marine worm, the swimming larvae has no sex. So if it settles down alone, it develops into female, that is 2.5 centimeter large, and if it lands on near or existing female, a chemical from her causes the larva to develop into a tiny 1.3 millimeter long male. So male lives as a parasite in the uterus of the female. While in case of second, that is an American alligator, that is snapping turtle and some other reptile, sexes is determined by the environmental temperature. In turtles, a temperature below 28 degrees centigrade produces more males. Above 33 degrees centigrade produces more females, and between 28 to 33 degrees centigrade produces males and females in equal proportion. So this shows the environmental sex determination. Now the last, that is determination of sex by the non-chromosomal gene. In case of bacteria, the fertility genes or the factors located in plasmids determines the sexes, and this mechanism is also called non-allosomic genic determination of sex. So this is the end of video in which you understood about the linkage, their types, discovery, factors affecting linkage, significance of linkage, sex linkage, characteristic of sex-linked inheritance, crossing over, their mechanism, resulting of crossing over, types of crossing over, factors affecting crossing over, significance of crossing over, sex determination, and their mechanism of determining the sex. So this is the end of video. Now the next upcoming video will be on the variation and mutation. So if you understood this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any suggestions, any queries, you can ask in the comment section below. Thank you for the watching.